Today, we are going to talk about matrix metalloproteinases, or MMPs. MMPs are the major enzymes required for the timely degradation of extracellular matrix components. The human genome encodes for 23 different matrix metalloproteinases. Each of these MMPs degrade various proteins that make up the components of the extracellular matrix. I have constructed a model that contains some of the components of the ECM and underlying structures of the epithelial tissue. These include the interstitial fluids surrounding the ECM, integrins that bind to the cellular membrane, fibronectin that exists as a protein dimer, collagen, which makes up the majority of the ECM, and a blood capillary that supplies the ECM and overlying tissue. But these normal physiological conditions are subject to change due to circumstances such as injuries.
There's a squirrel there. Where? Hey, right on the stump. Hey, give me some of your peanuts. Okay, here. Got him? Yep. Is the camera on? Yeah. Yeah, it's on. Hello, buddy. Hello, buddy. You want some peanuts? I can't believe it's not running away. Hey. Hello, buddy. You want some peanuts?
This is the closest I've ever been to a squirrel in my life.
That's insane.
Oh!
Good,
dude! Benny! Look, I'm bleeding!
Oh my!
This unfortunate episode has left our tissue damaged. The injury has destroyed the order of the ECM and perforated the underlying blood capillary. The tissue must be restored in order to prevent further damage and protect from pathogens. Growth factors, cytokines, hormones, and other signaling molecules are secreted by cells located near the damaged site. These molecules increase production of matrix metalloproteinase precursors. These precursors, or zymogens, begin with a C-terminal, or catalytic domain. This is bound to a zinc ion. The N-terminal of the zymogen covers the zinc ion. A cysteine is covalently bound to the zinc ion. Now that our MMP zymogen is complete, let's display how it becomes an active enzyme. The N-terminal of the protein is cleaved to expose the zinc ion in the catalytic domain. The sulfhydryl group on the cysteine associates with the zinc ion and the bond between them is broken, allowing a water molecule to bind to the zinc ion, activating the enzyme. Since the majority of the ECM is composed of different types of collagen, most MMPs are collagenases. The MMP is only able to cleave one alpha strand of a collagen triple helix at a time until the fibril is completely severed. Some MMPs degrade fibronectin, which is bound to the integrins in the cell membrane, and also contains binding sites for several other ECM components, which includes collagen. These degradative actions help with processes such as platelet aggregation on blood capillaries to aid in clot formation. These actions also release growth factors and other signaling molecules that recruit immune cells and other molecules to aid in the reformation of the ECM and other over and underlying structures, restoring the tissue close to its previous form.